Good afternoon and welcome to the 23rd episode of Genomics Gupshup. Genomics Gupshup is created to create awareness about genomics among the average person, clinicians and others who are interested in this great field of genomics. So today we have Dr. G.R Chandak, who has been a pioneer in this space. He's not just a MD, but he's also a PhD and has been doing a lot of research in the space of genetics and genomics. If you look at his scholar profile, you can keep on reading and it will not end. So he's currently the chief scientist and group leader at the very established and prestigious CCMB, that's the Center for Cellular Molecular Biology. Welcome to Genomics Gupshup, Dr. Chandak.
Thank you very much.
So I know that it's difficult to go through your entire great portfolio of things that you have done, but I want to start at the beginning.
Yep.
And I think we have one thing in common, which is that we are both from Bikaner.
Yes.
So do you think that being from Bikaner gave you something of an extra trait or extra something that has helped you in all your long list of achievements?
Well, that's an interesting question, I would say, because I never thought about it. So probably I have very vivid memories of Bikaner. I mean, I was born there, but then I never stayed long enough. I was rather brought to Calcutta by my parents, and then all my education has been there. But yes, we have been to Bikaner on a regular basis, as it would be during summer vacations or any functions and all that. So I only have very happy memories of the place and interacting with people. I think that the kind of the collectiveness or the interaction with family members, staying together, sharing their sorrows and joys, that was something which probably helped me develop good relationship, have large number of collaborators that I'm really very proud of. And second, I think that the sort of idea of doing something different I probably get from my father and from my uncle. They both, as during that time, had pretty difficult times, but I mean, both in financial terms and others. But I think they took up the challenge and kept on doing various things till we are really very stable. So I wouldn't be able to say that Bikaner was the one which shaped everything, but yes, in hindsight I think that interacting with lots of people, friends, family members, that makes it a little bit easy to get on with people.
So mostly I start some of my presentations saying I'm a Marwadi and that's it. I think people smile. I think because it's not just not just genetics, but it's also like you mentioned,
Yes.
the community, the sense of
Yes.
helping each other out and being
Oh
part
yes,
of the.
yes.
So that is what I was wanting to bring up a little bit. But and I remember Bikaner many years ago, and I remember how did you come out and actually to become a doctor and then this thing. Not an easy thing, at
Easy.
least at that point of time. I don't
Yeah.
remember there were that many people who studied that much. Right? So who inspired you to do that? And
So, as you rightly said that during that time education was less of a priority. It was rather business, and like Marwadis are known for their business skills, and I think world over, I guess. So I would say that I think probably my father, my uncle, probably because they had gone through such hardship in the absence of a formal education. So both of them were determined that none of their children will remain uneducated. And then it was those times when kids used to listen to the parents. Okay, so my father had ambition that he would have a doctor, a sort of a chartered accountant, and a management kind of a person in his. So we are three brothers, and so my fate was sort
you are
of
the destiny.
decided by my father, and I must say I was fortunate to sort groomed like that also. And I got very good teachers, I would say, biology, maths teachers. But those were, like say 78, 80 when I passed 10th and 12th
This was in
exam.
Calcutta.
It was in Calcutta, yeah. And but we had very good biology and maths teachers. But as I said, that my fate was already sealed, and so it was nothing that I made a choice. But I must say that I really wanted to become a doctor, and um, it was a very nice experience during my MBBS, MD, and subsequently also the fact that I'm able to use my medical knowledge to do some of these
So then from
research.
being a doctor, you became a double doctor.
Yeah.
You did a PhD also.
Yeah, I did a PhD. Actually, I mean, I must say that this is something very, I mean, peculiar. When I came to CCMB, I was an MD in biochemistry, and when I came here for a postdoc interview. But then I was told initially that, well, your MD is equivalent to MSc and your MBBS is equivalent to BSc. So I was I was first considered not fit to appear for that interview. But then somehow the directors thought that this is one of the few doctors who is coming over, let's try to catch him. So I joined as a postdoc, and then over a period of time, there are two things which led me to do a PhD. First of all, I was never trained, in any molecular biology. At that time, there was hardly any genetics taught, let aside any technical thing. Of course, I had a privilege of working with Professor Lakhotia, Dr. Raman in BHU. So I had some preliminary experience, but I realized that unless I do things on my own, it will be extremely difficult. And to be honest, I didn't have, at that time, serious plans to continue on research. When I came to CCMB, my idea was to spend 2-3 years, have good experience, and then go back, and start providing these kind of services, because at that time there weren't many places. But then when I came, then I really got, engrossed. Also, CCMB also helped because the atmosphere was so good and, infectious, I would say. People will talk to each other, no hierarchy, all the systems open. So then I thought that if I do a PhD, that means that will be something which I'll be owning up also. So I decided to get onto a PhD, and again, Director CCMB and others were very supportive at that time. So yeah, you can call as a double doctor.
Yeah. So after that, you've been involved in many different areas, right? I saw that you've done in diabetes, in many other
Yes.
many different areas. So which has been your most I know it's hard to say that when you've done multiple things, but how has your how did you get interested in so many different research areas?
Yeah.
And what has been the most exciting for you?
So as a physician, I have always been excited to give something back to the society. So although I have done, I feel that there are many interesting things that I have been able to do, but still my first love remains molecular diagnostics, genetic counseling, prenatal diagnosis, because of the fact that this helps us contribute to the society. And I think that any research, anything that you do, in my opinion, it should find some application somewhere. If it is agriculture, you know that it is a very large implication. Not that I am saying that basic research is not important, that's extremely important, because unless we understand the basics, we really can't move forward also. But that one thing still remains my passion, and actually, if I recollect my 25-30 years, I came here in 94, so close to 3 decades journey. 94 I came to CCMB, and then right from the first day, actually, Dr. Lalji Singh was my mentor at that time, and right from first day we struck note on one thing, that we have to develop a state of the art diagnostics facility, everything. And he was over ambitious, much more ambitious than me. He would always say that, well, there are 6,000 genetic disorders, we should develop facility for each and everyone. And I used to just think that, oh, I mean, this man is talking about 6,000, we are not able to do for 10, 15 because of technological limitations. But see now, it's possible. It's possible to discover new diseases actually. So, just in 3 decades' time, this whole thing has come that way. But I would say that it was very nice because I could develop this facility at CDFD. I was one of the founder faculties of CDFD, and it's always happy to see that it's really gone very well. Then at CCMB also we do in our own way also. So I'm very much excited about that. The other thing is, which was my as research my first love, which was like looking at chronic pancreatitis. I worked on that for almost 15 years and I learned a lot from there. I must say that that whole journey, along with Dr. Nageshwar Reddy and Dr. G.V. Rao from Asian Institute of Gastroenterology, that has shaped my career to what I have been able to do. And in just one or two lines to say that, two things which we identified particularly was that the genetic basis of Indian diseases are different than Europeans. And second thing was that genetics is not the boon or the bane of all the diseases. That led me to complex diseases like diabetes and many other diseases, where we realized that other than genetics, it's your environment or lifestyle or nutrition that actually shapes the clinical picture that you will finally have. So I'm really very thankful to have chosen these two areas at the initial stage of my career.
Yeah, I mean, I think that's one of the things that we keep telling people, right? We're not doing understanding of genetics to say it's good or bad. It is more to be able to say how do you optimize or how do you understand all the other risk factors that are there. But in that, you mentioned pancreatitis. I saw there was a Hindu article on, why do Indians get more, there's a difference. So you had mentioned some of the gene names, like SPINK1, and you said that Indians have more of a higher frequency. For an average person, what does that mean? Like, does it mean that we are more likely to get pancreatitis, or what?
Yeah.
Maybe you can explain to.
So this was the initial years when we discovered this. We discovered that one of the genes, cationic trypsinogen, present in Europeans was not a risk factor in Indians, and we discovered this particular gene also. So what we also found that this gene is, every gene has a risk variant and a normal variant. So the risk variant was present in about 30 to 35% of our population, compared to about 8% to 12% in the Europeans. And at that time, the disease was largely considered as a few genes would be causing the disease. So because this cationic trypsinogen in Europeans was causing the majority of the disease, and we had found SPINK1, which was having a very different genetic structure. So we hypothesized at that time, and again like going by the because these are complex diseases, which means that the risk runs in the population itself. It's because of some additional factors that one develops the disease, like it is for diabetes or other. So the bottom line is that if a particular risk variant is present in a higher proportion in a population, that means that the disease is more likely to be highly prevalent. And that is what was the hypothesis at that time. And I still remember that when that was announced, I think it was, I'm forgetting the name, but it was Somshekharan, I think. He was the science journalist in that, and he asked me that, so can you tell people that if you have SPINK1, then what is the chance of developing pancreatitis? And that question I have been facing since then actually. So I used to say that, well, these are early years or so, and actually we were proven true, because today there are some 15, 16 genes which are known to be associated with pancreatitis, but that technology allows us to investigate that. Of course, I mean, as I said that these all diseases are also dependent on lifestyle, because just having the risk variant doesn't make you develop pancreatitis.
Same with obesity.
Absolutely.
Many other
diabetes, cardiovascular diseases also.
So one of the things that I find fascinating is how people come up with these gene names.
Right.
I mean, how do you figure out, like, why is something called BRCA, why is something called SPINK, why is something called this thing? So, like, you probably deal with this on a day-to-day basis, right?
Yeah.
Is there some method to this madness of, like, or is it just depends on, Dr. Chandak has, found this out, so he'll name it,
Interesting question. I mean, as far as my knowledge goes, that people usually do. I mean, if a gene name is usually decided by, gene name is, I mean, either decided by the protein. Again, you would say then how the protein name is decided. But as far as I understand that any of these nomenclatures, they're essentially supposed to give an idea about the structure or the function of that particular gene or the protein. Like say when I say SPINK1, without going into details, but it stands for the serine threonine protein kinase. So the moment you see this name, you know that well it's a kinase and there are these two amino acid residues. Same thing holds for say hemophilia A gene. I mean it's very simple. That means that this is the gene which leads to hemophilia. So there are various ways I think it has come, either say the deficiency causes a disease or that it leads to a particular function. I think that the names after scientists, I think they are more common in some of the species, like any infectious organism you find, then you put those kind of. But as far as I remember, gene names are usually mostly protein based, and then for a specific function. Of course, I mean now we have so you must be aware that when there is nothing, then they would just say ORF, open reading frames. And then as the function would get more and more correct, now we have all long non-coding RNA to whatever, so many are there. But I mean these all are intended with the purpose that, I mean, looking at the name itself, some information is gotten about the function or the disease that it might cause.
So one of your other areas of research is methylation.
Right.
And I remember having this conversation with some people about methylation. But for most people, they don't understand, right? Like, what is methylation, right? What does it really mean? So, when you are studying methylation, what are what do you study in that? Like, and can you do it with, how do you actually use genomics or genetics to study it?
Right. I mean, I will try to simplify it, I mean, although this is a very age-old mechanism. So, if I have to say methylation, then it's just a simple reaction, where methyl group is added to a substrate and the product which is methylated essentially. But this methylation has been one of the most important regulatory reactions. I mean, we know that, stone methylation or say any methylation at a particular, CPG island, which is sort of present in the promoter region, it's a regulatory region. So methylation essentially is a phenomenon by which the gene function is regulated. That's the simplest way that I can say. And how do we contrast it with a particular structural change, which we usually call as a mutation in a gene? Is that a structural change will usually lead to a replacement of amino acid, like it happens in sickle cell anemia. In the protein there is a glutamic acid, but because of some mutation, lysine becomes, so the whole property of the protein changes. So similarly many other changes can happen because of this. Sometimes because of a structural change, the protein may be truncated. It may become small, like it happens in hemophilia. So there is function of the protein is lost. It loses, it remains only 10% active or something. Whereas methylation in contrast, if suppose if we say methylation, that means it will influence the RNA level and then subsequently the protein level. That's the kind of relationship that usually thought. I'm not going to make it very complicated, but suffice it to say that if there is a methylation at a particular regulatory region of a gene, then it is likely to influence its levels. Which means that if I needed 50% level, or say 50 milligrams of a protein to do certain function, I'm now going to have 10 milligram and all that. So that means it's likely to cause the problem. What is equally important is that the structure of a gene you can't change. It's what you are born with, and that's what makes you what you are. But then methylation is something which is dynamic. It is dependent on various factors. But for the sake of simplicity, for larger audience, it can be influenced by as simple as your diet or as simple as any kind of vitamin supplement that you take. Say for example, if I
B12.
talk about B12, yeah, that's a, so it's a, and many of the people wouldn't think that B12 is important, but it's a kind of a B complex group of proteins, and it has attained big importance in last few years. So if you take B12 or say any other dietary changes, then it is likely to influence the methylation of certain genes, and which means that it will influence the protein levels of those genes. And the fact that the genetic structure cannot be altered so easily, but the function of a protein due to methylation can be altered so easily, that's why there is a lot of excitement. And this area of this methylation, it's one of the components of epigenetics which has come up. So genetics means your gene sequence, your genetic structure, and any aberration leading to either a clinical or an aberrant phenotype. Whereas epigenetics means that something which is not genetic, above genetics, so a change which can occur without any change in the sequence of DNA is what we would call as epigenetic changes. And people might have heard, I'm just mentioning these terms like miRNAs and long non-coding RNAs. These are also sort of epigenetic modifications which can happen. So in nutshell, while genetics determines what you are, but probably epigenetics determines that how your life course, and how your clinical profile or any kind of disease may occur.
So essentially, I think when you mentioned initially, it's neither a boon or a bane.
Yes.
I think it's because of all the environmental.
Exactly.
And I think the greatest thing has been that now you can actually measure these levels as
Yes.
well. Right? And I think one of the areas like the B12 thing, there are I think methylated versions of the of the B12.
Yes.
I think supplements also that now I think are being sold in.
Yeah.
So I think it's fascinating how much information we've been able to gather over the last so many years.
Yes.
And the people should realize that all of this information is a guide. It's not like it's not a destiny, right?
Yes.
And I think that's one of the biggest things that we want to get people to understand, that anything is there so that it can help you to get to a better state without being compromised in terms of your quality of life.
Yes.
So you're also working on gene-nutrient interaction. Right? So can you tell us like a little bit more about how this research helps the average person? I think we mentioned a little bit about B12, but are there other areas of nutrients and genes that that will have a huge impact on a regular person?
Yeah. So I mean, this also is something I would say which I learned from my early career, and I would say more than success, the failures. Because when we started working on diabetes, we that Indians developed diabetes at least 10 years earlier than Europeans, and then Europeans develop diabetes when they are obese. Obesity is considered to be important factor. But that Indians are not obese, to say the least. But then they have a very different kind of obesity. They are centrally obese. So they have lots of fat around the paunch and all. And I'm sure the audience knows that people with a paunch around have more trouble or so. So it's like we are usually taught that it's your genes which decide the way you look, the way you walk, the way you behave, the way your protein function is there. So when we go like that, that means that this genetic constitution determines everything. But as I elaborated earlier also, that it's not so. And we have always been excited because, I in particular, being from a medical background, uh, I would say reasonably understand the clinical heterogeneity. So the work on calcific pancreatitis also started from there, and then we did find that there are different genes which are causing the disease. So same thing we went with diabetes also. We thought, well, early onset, lack of obesity, Indians are more insulin resistant, there must be something genetic. And then over almost several years we kept on working, and we couldn't identify a novel gene for diabetes in Indians. Of course, we found very different mechanisms, and again, I'll not go into the details, but from one of the studies which we did, and there was a gene which was called FTO, which was identified in Europeans, obesity gene. That's where my first movement towards this epigenetics or nutrition started, because what we found that FTO would work through obesity to develop diabetes in the Europeans, but for us it was an independent risk factor for diabetes, so it wouldn't really work through obesity. And that is where we thought that, well, there could be a different way that this FTO might be regulated, and that's when we started moving towards these nutrients and other things also. Now it was, on a different perspective. One question which I started having about 10 years back was that, okay, even if you find that somebody has got a risk factor, say some diabetes risk genes are there, but what do I do to them? I cannot change the genetic constitution of the individual, and by the time we find out the individual is already in 20s or 30s, so what do you finally do? So essentially, the whole thing goes back to your lifestyle modification, your walking, your nutrition and other things. So that was really a big dilemma about 10 12 years back, and it was like leading on a road which is already closed. You, even if you find hundreds of genes or so, you have nothing to hand over to the person who is at a risk. So what am I doing? Then I ask myself the question that I am only labeling an individual at a genetic level. If there is a family which has got 5 diabetics, then they know that they are going to, the sixth one is highly likely to develop diabetes. What am I doing by doing genetics to that person? Except that we motivate them further for lifestyle and other things. And that is where I came in contact with people who work on programming, Dr. Yagnik amongst them, the most important person, my collaborator, and I work with Dr. Kumaran, Dr. Krishnaveni, and many others actually. So what I learned from there is that, so since you asked about this, where the nutrition can come in, what I learned that it is again, not only your genetics, but then your environment, which can be as early as the intrauterine environment, which means that the mother's nutrition, which determines the nutrition around the baby for the first 9 months in the womb, that determines many of the things. And again, to cut a long story short, it's very clear that the mothers who are diabetic during the pregnancy, this is an age-old observation, that the mothers who are diabetic or have high glucose levels during pregnancy, their kids are born big, because glucose acts as a nutrition for them all the time. And these kids, once they are born, they are at a very high risk of developing diabetes in future. So this was one of the earlier observations which said that, well, mother's nutrition has programmed the fetus to develop diabetes in future. And this field is called, earlier it was called fetal programming, now it is called developmental origins of health and disease. Because this has not only led to diabetes or others, it's also
Many
influencing many other cognition and all that. So this is what we have been looking at, and we have used both genetics, a huge amount of genetics data. I work with some 5-6 cohorts, each of which have about 500-600 mother, child, and father pairs, trios, and then their data at every 5 years, 6 years. All these
These
data
are
have
full
been.
longitudinal.
This is a full longitudinal cohort of 2 decades or so, and currently all these kids are at about 20 to 25 years of age. So I'm really thankful to these collaborators, even international collaborators, Dr. Caroline Fall in particular, and many others. So now our understanding is, just to summarize, that while it's important that your genetic constitution has to be correct, but that is mostly important for rare diseases, which means if I have a mutation, I may develop sickle cell, I may develop beta thalassemia and others. But what is equally important is the number of other kinds of variants, which then in interaction with early life environment, like I said, mother's nutrition, or even at a later point of time also with your diet, your nutrition and lifestyle, that can finally decide the development of diabetes or say cognition problems or other things.
Absolutely.
Sorry for this long answer.
No, that's precisely. I think that's the area that
Yeah.
we do a lot of. We've been. I think one of the biggest thing is telling people that the reason you're doing this is not to, like you said, it's not a destiny.
Yes.
It's more about helping and guiding them.
Yeah.
So, for instance, genetically, I'm, very prone to diabetes.
Right.
I have a family history also.
Yes.
But you manage the environment and your nutrition
Lifestyle.
and lifestyle.
Yes.
And today my HbA1c is 4.9. So it is.
Wow.
So I think I've been able to keep that in
Sure.
in check, and I think that's what we want to tell people, that yes, it's true that you have a slightly different genetic makeup, but
Right.
it means you might have to try a little bit harder in
Harder.
some cases, but
Absolutely.
you
Yes.
can do it.
Yes. Yes.
And the same thing with many others. I think I discovered B12 deficiency the hard way.
You had trouble.
No, so I think a doctor almost was going to operate on me, thinking I had carpal tunnel syndrome, saying that you work so hard on your
Wrist and all that.
wrist and you're working all the time, and therefore we need to operate you. Luckily, I went to another doctor, and he basically told me that get a B12 test done, and that's when I found out. This was many years ago, more than So I think, and later on when I did the Genomepatri, I found that it showed over there that I had
Particular gene.
Yeah, that I had the
I see.
risk over it. So if I would have done it already, I would have known. But
Sure.
okay, ab to pata chal gaya. So that is one thing that we find.
Sure.
So I know you have many other research interests. Any other thing that you find exciting or you're working on right now? I read about a very large cohort that you are working on right now, some 53 lakhs something.
No, I mean this is I think my journey is now pretty clear that I started from single gene disorders, worked on diseases which had both genetic and possible environmental component in pancreatitis, that then moved on to common diseases like diabetes, and then tried to understand the time point at which the risk develops so that we can intervene at that point of time, and have tried to understand the molecular mechanisms of that how early life exposures influence epigenetic mechanisms, so that we can guide any individual right at birth or just after birth, because early intervention is always the sort of idea. So from while that continues as a big sort of part of my my lab work or everyday work, I have now taken advantage of my earlier experience with these monogenic disorders. So now I work on two particular disorders. One of them is hemoglobinopathies and other is musculopathies. So I long back, it was I think in 99, 2000, I worked on sickle cell anemia, because in some tribal populations of Vizag, I think it was not Hakipiki, I forget, some release. they had sickle cell anemia, and I was somehow referred by some University of Andhra University. And what we found, that was a small study, but what we found that the Indians had a very mild sickle cell phenotype. You know that Africans have very severe phenotype and they die early and have lots of complications. So to just to put it in nutshell, that we at that time we were the first ones to find out that, well, sort of, I wouldn't say Indians because that was an Andhra study. We found that sickle cell patients there had high fetal hemoglobin level, and that was because of a particular genetic variant. I mean, we looked at many variants, so it was a haplotype, Arab Indian haplotype. And then I worked for 2-3 years on that, but then I was consumed with this pancreatitis, diabetes, and all that. But I didn't forget that when this whole thing was going on, I started working on sickle cell anemia again. And actually, CSIR came up with a CSIR mission mode project on sickle cell anemia, and I was privileged that they sort of gave me the opportunity to lead this. So just in nutshell, I will tell you, because this is something important if we want to cover a disease which is prevalent in a population. So usually when we talk about genetic testing and other things, we are waiting for something to happen. When it happens, then we test and then we go back and try to prevent the disease. But for diseases which are reasonably prevalent, like say sickle cell anemia, it's not that common a disease, but then in some areas, I mean 6 to 7 states actually, not, I mean some areas, it's big, like Maharashtra, then Odisha, then Gujarat, then Madhya Pradesh, all these 6-7 states, that whole central belt, sickle cell anemia, the carrier frequency is about 10%. And disease frequency is 0.4%. Okay. So what we thought at that time, I'm again my close friend and colleague Dr. Patra in Chhattisgarh and Dr. Deepti Jain in Maharashtra. So we started to work together, and it's a big program, but I won't go into much details. What we did was that we decided on a population screening program. So in state of Chhattisgarh and Maharashtra, and then in Madhya Pradesh, we started screening for school-going children, and then in hospitals, the newborn screening and all that. So we have done close to 30-35 lakhs of children screening, and also several thousands of patients, their families and all. It's the 6th year that we are running in them. So what we have been able to achieve is that we realized that there is no point of care testing at that point of time. Whatever is there is all available from abroad, and that would cost about 200 rupees per test. And then we realized that if you want to do a genetic test, then you have to still do several things. So what we developed is small PCR based approach, where you just take a small drop of blood sample and you can do the whole genetic testing, and that's very cheap as well.
You're doing this for the child, or you do this for the parents? Like,
so,
is it done more as a carrier screening, or
so our approach is, I mean, till now we were mostly going to school children, because that can be, because it's not possible to do on a complete population base unless you have state support or center support, which is what now we are going to do. We are in touch with ICMR and others. Now we started from there, but once suppose a child is identified as a carrier, then we go to the family. So we have I think some 6,500 families, data, pedigree charts and everything. So we then counsel them. There are different kinds of counseling that is done. Then we offer them prenatal diagnosis, all free of cost under this CSI sickle cell anemia mission. So overall, at the end of this, almost at the fag end of this project, what we have been able to do is have a comprehensive screening plan, that how do you approach a sort of a village or a district or something. How do you get to a child? How do you screen? What do you do once a child is positive? How the treatment is taken care of? How genetic counseling is done? So that sort of a plan we have done, implemented in Madhya Pradesh also, and we are quite confident about that. And second is that we also did quite a bit of research along with my colleagues from other CSR labs. Looked at this, as I was saying, about hydroxyurea. Hydroxyurea registration as a drug for use in sickle cell anemia, and we also try to investigate the new drugs which can come up from the herbal background or any other chemically modified entities, and then also to look at a final treatment for sickle cell anemia. I mean, you know that this is only because of one single mutation all over the world. So using genome editing, using CRISPR-Cas to basically correct that and reinfuse the modified cells, that sort of thing. And I'm happy to say that we are on a good track on that. And hopefully we will have something, but that's a longish procedure to go. But regarding this screening and all, I'm extremely happy and proud to say that this program has been to great satisfaction for me, because you identify children and it has an impact also. I'll quickly talk about second one, which is musculopathy. So we have worked on Duchenne muscular dystrophy and spinal muscular atrophy. There is an organization called Indian Association of Muscular Dystrophy. It's actually Solon based. Through that and their chapters, I have interacted with large number of muscular dystrophy patients and all. Again, this is a disease where there is a big social stigma also, because this disease passes through the females and affects the males. So we have worked on various aspects,
social aspects.
Oh, lots of social aspects. I mean, I would have shared many interesting stories where a lady came for getting the carrier status determined and getting a prenatal diagnosis done, completely hiding it from her husband's family. And one time the husband's family, literally searching for the lady came to CCMB, and we had a tough time. We had to hide the lady inside the toilet and just say that there is nobody, we don't even know, and stuff like that. So anyway, that was on a lighter, but obviously very, very serious, kind of situation for many of the diseases where this happens. So I've been working with these people, and our motto has been that, again, this is a disease where there is no treatment, no definitive treatment. So there are research groups which are working on this. I'm not involved in that. But my motto there has been that it's a disease which can be prevented. While you're developing the cure, why not look at preventing the disease? So we are trying to look at the families where there is a disease, and then go back to the females, get their carrier status known, and then prospectively go and keep a track on these women, that where whenever they are planning for a pregnancy or they become pregnant, then so it's like, rather than bringing those women to the doctor or to the clinic, it's we who go to the family, keep a tab on them, that what's the plan. So that has been also quite helpful. And I recently actually Honorable Prime Minister also had visited IMD, and he actually put it in Man Ki Baat also, that muscular dystrophy is such a disease. So following that, there is quite a bit of awareness and excitement about this. So I was also asked by these Man Ki Baat people that what is the disease we don't know. So I was very happy that these efforts, it's a long-term efforts. I think somewhere these diseases which are sort of, I call them as commonly occurring rare disease, because now rare disease means people think that it's rare. Actually, it's not. It's not rare. I mean, if you talk about 0.4% sickle cell, which means you're talking about 4 per thousand. And considering our 1.6 billion population, how many are we talking? Muscular dystrophy is something like 3 per 10,000 or so, but that's males. Carriers will be even more high. So these are some of the diseases, I think, where a prevention strategy can be there till we get into the treatment.
I think so that people need to be more aware.
Aware also, yeah. I mean, I completely agree. I mean, that's where I think it's not possible for one individual or a couple of individuals to do. Yes, I feel yes, the government has to come, or some sort of a. I think that I personally feel that some sort of a public corporate partnership should be coming up. Maybe corporates also need to get into some of these social aspects, because the bulk is so much that I mean it's massive. Hope that at some point of time we will be able to knock and get something
I think
done.
it's slowly starting to
Yes,
happen a little
I
bit,
agree.
because
I agree.
even when you look at like things like your screening for cancer and others also,
Oh, yes.
I think now corporates as well as insurance and others are also at least
Yes.
waking up to the fact that
Yes,
these are things
absolutely.
that
Yeah.
these are easier things to do, I think, and I think the Nidan Kendras and others that have been
That's
created,
coming from DBT
yes,
actually. Yeah.
those
I think
are all,
that's a very good effort as
yes.
well. Yeah.
So I know that we can, I think if you go through your list of this thing, we can continue to talk, but I know that we've come close to an hour, so
Sure. Yeah.
is there anything else that you'd like to talk about, or I'll move to the rapid fire?
No, I think that it's a very exciting time. I feel looking back at when I moved in this field. Since then, there are a lot of pluses. First of all, I think first and foremost is that the technology has become so powerful. I would say that I mean today data generation is not a problem. Data analysis is a problem. You have mounds and mounds of data. I mean, since you are also into this, and we are also doing a next generation based sequencing, and you are preparing mounds of variants of unknown significance. So I think that while this is important, it needs probably a consortium or a collaborative approach that clinicians and data analysts and technical team, they take up this as a challenge. And I'm sure that India is a huge place, and it's a place of golden opportunities, genetic diversity, ethnic diversity, disease diversity. I think that a sort of an effort is required where the motive should be to help the society, the patients, and I think India has a huge potential in that regard. Second is, I think that there's a huge interest amongst the clinicians also. I mean, I remember that when I came 30 years back over here, I was said as the crazy person, and people used to joke that iska practice nahi chalta hoga, that's why he has come to this field. Otherwise, who will come at the 3,500 salary at the age of 35 years and spend time and all that? Actually, I was I was an outcast in my family also for quite some time, because Marwadis are business people. Then a doctor, Marwadi doctor means should be running in riches. So, but anyway, I think, but it's really very interesting now. Now there are opportunities available. Now there are medical genetics courses, clinical genetics courses. Funding agencies have come forward. We ourselves run a training program of 2 weeks for medical students. So I think opportunities have become huge, particularly I would say for the medical community, the awareness. And one more thing is, I feel that clinicians have become a little more humble. Humble in the sense that they now probably think of talking to other stream.
Yes.
I would say that realizing their lacunae also. I mean, it's important that all of us realize what we don't know. Rather than trying to acquire all the time everything, it's important that just cross that one line, sort of Lakshman Rekha, and go across and find that. So that's something which is, I think, very very important, which has happened. And lastly, I think that society has also become very aware. I mean, these are the three important pillars, and I think society also is now very aware. Of course, I mean, many times they ask all kind of other kind of questions, but I think that the people on the other side of the table have to be able to satisfy them. So I'm saying that people like counselors or clinicians or the other people, that's how it has to be done. And last but not the least is that the research part of it. I think I would say that that's the hub kind of, a basic research with some inclination, and then all these things as a spoke is what. So I only tell that, for the common man, whichever way you can, either be aware or get into clinics or biology or anything, very very exciting time. So I think that's what.
More medical students should get into
I agree. Yes, yes. No, I mean, I was telling you that there was a bunch of 20 medical students, and you won't believe. I mean, even after their course was finished yesterday, they spent almost an hour with me. Five of the students, three were from Osmania and two from my own old medical college, Arjikor, and the kind of questions they were asking, I was very happy. I said, even one of you come into this, I'll be very, very happy. So I think it's exciting time for this particular.
So do you think we should make some changes in courses for medical students and also maybe changes in courses for people who are doing masters in like biotech and others so that they also understand
Yeah.
how medicine works
Yeah.
more and vice versa.
So I think changing courses and other thing is a little difficult job. I because sorry, because the primary objective of a medicine guy is to treat, and in India we don't have too many doctors. So every field that there's a problem, there's a lacuna, there's a deficiency of doctors, and same holds true for the other side also. While many people graduate and postgraduate, but most of them actually go abroad because there aren't much opportunities over here. Not, I wouldn't say opportunities, but the right opportunity people don't get. So I am of the opinion that both these sides should be with their curriculum, but there should be some kind of bridging. I wouldn't say courses, but say some sort of a bridging
Finishing
lectures
courses.
or finishing courses. Some of those 3 months, 6 months of modules, which can, like say, I would tell you this 2 weeks, course which we did. We found that they were extremely interested in holding the pipettes and asking all kind of questions. So follow them up with another course at the end of 5th year or something. That would be. But yes, what you suggest is important. There should be more of cross talk. Only thing is that if you like put lots of material in that same time duration, it may be a bit difficult. So kind of of all the eggs in the basket, just allow or pick up some of them to get onto this.
We are actually thinking of at least collaborating with some institutions
Sure.
to see if things like pharmacogenomics or other things that can be done.
Yeah.
I remember many years ago, 20 years ago, when we started Osimo, it was so hard to find people. So we did these, I think, university collaborations with the US. We did some training and got people, and in many ways, I think we were like one of the early. We were able
Yes,
to.
I remember that.
This thing.
Yes, yes.
Now I think at least it's good because there's enough people who are
Yes.
available and so on. And I think maybe we need to do that, both for genetic counseling and
Yes.
also other areas that can.
Sure. Yeah.
So I know I've taken more time than I.
It's always
thought
a
of
pleasure,
it,
actually. Yeah.
So now it's a quick rapid fire.
Oh my God. Okay.
So what do you prefer, books or movies?
Both actually. I'm fan of both. Of late, I've become a bit mobile guy, so I look at books, movies, everything on that only.
Which is any favorite?
I'm a Hindi guy. I don't like English books or other Prem. I read Prem Chand and Sharat Chandra and all that. These days, I'm reading a bit of Robin Sharma and I don't remember these foreign names of authors, but these are some of my favorites.
What's your most productive time of the day, morning or night?
Both.
Both.
You have chosen these two, early morning and
Yeah,
late night. So yeah. So you can consider late night to early morning.
Okay, so that's your time. Your night to.
Yeah, I think I'm a poor sleeper actually. So if I wake up and if I feel fresh, then I just start working.
So it doesn't matter what time it is. You can. If you're
Daytime goes more in administrative activities, to be honest, discussing with students and others. But yeah, evening after 9:30 till say early morning is a good time for me.
What's your favorite food?
Food, yeah, Rajasthani food. Unfortunately, I can't have most of it now. But yeah, jalebis and kachoris. These are my most favorite food. Of course, I'm very fond of Rajasthan. Yes,
Yeah.
I'm very fond of Rajasthani food like khichda and all those kind of things. Gatte ki sabji. It's another matter that I have had my quota finished very early on. Yeah, just on these festival occasions on that time. My mom will not leave me unless
Unless you get
she
that.
I get that I take it.
So, any New Year resolution?
See, I'm going to be out of active government job in 6 months' time. My resolutions have never succeeded, but that doesn't stop me from making resolutions.
Yes, exactly. That's what they actually,
Yeah.
that was my next question. Resolutions are to keep or to break.
Yes, absolutely. No, so I mean it's something as I do every year. I take a stock of what we have done in last 1 year. So what I do is that I request all my colleagues to make a New Year resolution because I am what they are. So I ask all of them that what is it that you have achieved in the last year, what is it that's pending, and what new things you have. And then, of course, I also look at it that what is it that I wanted to or so. So I think during now I want to spend a little more time on societal things or so. Continue with my research because that is something which which is what keeps me motivated. Also. But I think that in during these years and next few years to come, I would focus on looking at more of these epidemiological programs, looking at these intervention programs and other things, and also try to translate my basic research stuff into something which can help the society. Just to give example, we now are working on developing a type 1 diabetes genetic score. We have developed one and we have published that also. But I am working more on that. So both simultaneous things is what I would like to do.
So that's an outstanding career that you've had. You don't look like you're ready to retire already.
Yeah. No, I'm not going to retire for sure.
Yeah.
As I said, my active.
Yeah.
Hopefully,
So,
I'll be able to do more things that I really love. Of course, I must say that I've never had that regret.
So
CMB
why
has
is
given
there
this.
like a limit to government service? I think.
I mean, I think that somewhere, I think government also is, sort of bound by
Some rules.
certain rules, and I think they also need to put a stop probably somewhere. I think that it's a good time to sometimes take a break,
Maybe do something else.
maybe do something else or so, and those who are passionate about one thing, I'm sure there will be opportunities from them. But I felt that this was a good time point for me also, not to take a break, but then just maybe take a detour, look at things, and of course, when you turn close to 60, then you have your family and other things. So I thought it's a good thing to say that, okay, how many years you have ignored your family, so you better now spend time with them.
True. So that was great. It's been an absolute pleasure to have
Thank you
this
very much.
Genomics Gupshup, and maybe we'll have a second round of discussion.
Well, I hope. So, thank you very much. It was nice talking to you. Pleasure. Yeah.